a professora aqui, a Emelie Katarina Svahn, Leão, né? Que é um prazer enorme tê-la aqui, porque eu já vi ela falar umas vezes anteriormente e eu achei muito interessante o trabalho dela, incrível realmente, muito interessante. E acho que tem alguém falando aí, Lu, vai ter que fechar os microfones, né? E ela atualmente ela é professora adjunta no Instituto do Cérebro da UFRN, né, em Natal, né? Eles têm a sorte de ter a tia trabalhando lá. E a formação dela ela fez um doutorado em neurociência na Australian National University, Austrália, né? E depois ela fez um pós-doutorado em Uppsala, né? Na Suécia, ela é sueca, e ela de 2008 até 2012 ela trabalhou, ela fez esse pós-doutorado num laboratório de audição também, né? Mas basicamente ela vai falar para a gente sobre o zumbido, tinnitus, né? Que ela tem trabalhado nos últimos anos. No e-mail que todo mundo recebeu também tem um pouco sobre as técnicas que ela realiza no laboratório dela, que ela colocou, né, que estão aqui, que eu acho que é bem interessante para todo mundo. E o fato de que, na verdade, é sensacional ter uma pessoa na área de audição que é muito subrepresentada, eu acho, no mundo e no Brasil também, né? Então, é muito bacana a gente ter essa oportunidade de escutar aqui. E para as pessoas interessadas nesse tema, né, acho que é uma pessoa excepcional, pesquisadora excepcional. Então, que é um grande prazer receber você aqui no Programa Avançado de Neurociências, finalmente, e da Suécia, por acaso, agora é da Suécia, né? Então, podemos, se você quiser compartilhar a sua tela.
Fazer direitinho.
Ok.
So thank you so much for that very very warm welcome to the talk. Let's see if I can just decrease the size. Maybe not. Yes. So this talk will be in English, and I'm going to tell you a little bit about the physiology of tinnitus or tinnitus, depending on if you learned this word in America or England, and the therapeutic strategies for the treatment of tinnitus. So as you heard, I'm currently, or I will stay to be a professor at the Federal University of Rio Grande do Norte, Instituto do Cérebro, and I love working with these people. So many of you might know about tinnitus, this phantom sound that you can hear without an external source. I hope you're not suffering from it, but it's kind of common, so I wouldn't be surprised. So tinnitus can be this loud, high-pitched tone, this beep you can have in your head all the time, and you can not get rid of it. It can be or it can be of different forms, but it's this zumbido that you have in your head. Most cases of tinnitus are related to hearing loss, but they're also highly correlated with noise exposures. And you don't have to have a hearing loss to have tinnitus. You can have noise exposure. This can be from like working in a factory or being at war and having grenades exploding or different kind of loud noise, but it doesn't need for you to lose hearing. You can actually just have an increased activity that leads to tinnitus. And unfortunately, we can already see this in the young population. So 2 studies that came out, both of them actually in 2013, have studied teenagers. So one in the US showing that nearly 5% of teenagers already have chronic tinnitus. And then the second study was from Belgium, and they reported that teenagers up to 18% already had chronic tinnitus. And this is very unfortunate, especially as the Belgian study also saw that the teenagers were not really aware of the risk of tinnitus and wasn't aware of protecting their ears from loud noise, recreational noise from listening with bad earphones or going to loud concerts and so on. So the problem with tinnitus is that, first of all, there's no cure. And there are not so many really successful treatments, although they are treatments, but they basically treat the secondary symptoms of tinnitus. And this can be difficulty to hear or to focus. If someone is calling you from a distance and they're like saying something and you in your head have this beep that's very loud, it might be hard for you to hear what they're saying. It might also be very hard to study, to focus, working or reading or something when you're constantly disturbed by this beep. This can of course lead to bad mood. You can feel stressed, especially people that have tinnitus that comes and goes, that stays for a couple of weeks and then it disappears, but then it comes back. This is like half chronic. These people can be very stressed about when they will get the tinnitus. And this can lead to anxiety, obviously, if you're worrying about how will your tinnitus interfere with your performance. And as with everything that's stress and anxiety, it can lead to depression. And some studies have shown that people with bothersome tinnitus, about half of them also have clinical depression, actually, as a direct cause. And as we all know, depression is now one of the real problems in the world. We have more than 300 million people of the world suffering from clinical depression, clearly decreasing the quality of life. And if already teenagers are getting tinnitus, you can just see that that will affect them throughout life, possibly. These treatments I mentioned, they are basically focused on decreasing the symptoms. So cognitive behavioral therapy or mindfulness, they are important for trying to deal with it, to not feel so stressed or not have such a bad mood. They work a lot on having acceptance. If you accept that you have tinnitus and accept that this condition will not hurt you, it will not damage your brain, it's just there, then it's easier to deal with all the other secondary symptoms. You can apply sound masking, which can be different auditory devices. You listen to another kind of sound that makes the tinnitus kind of fade in the background. Works for some people, not for everyone. But then there are two, at least, treatments where they actually focusing on the neuronal activity, like the actual creation of tinnitus, and that is transcranial magnetic stimulation or transcranial direct current stimulation. So there were two big studies made from some European framework where they looked at a lot of different neurological problems, and they looked at guidelines for when or when not to use, for example, transcranial magnetic stimulation. And they found that there's the possible efficacy of treating tinnitus or auditory hallucination with magnetic stimulation. So that's interesting, but possible shows that some patients have an effect, some do not. Some have an effect for just a few minutes, some for a few hours, but it's still very much early days for this to whether this is having an effect or not. A little bit more recent study focusing on this direct current stimulation, where you actually place electrodes onto your head and you have this dipole and you apply a current onto your brain that will go into the most surface area of your cortex. Here they show that it's a probable inefficacy. There's absence of clinical effects. So there's not much we can do to focus on trying to control this excessive neuronal activity, unfortunately. And that really shows that we need to study this much more. So first of all, just to understand a little bit what parts of the brain are dealing with tinnitus. So here is this schematic from a review showing that there's both auditory and non-auditory pathways involved in tinnitus. So here in the bottom you can see a schematic of the cochlea, obviously where sound is translated into electrical signals. This first enters our brain in the cochlear nucleus, and especially the dorsal part of the cochlear nucleus is important for generating tinnitus. this part is also interesting because it has a somatosensory input. So for example, wind blowing on your ear or vibration in the ground, different kind of somatosensory input that can help explain to the brain where sound is coming from or so. So this double type of input makes this dorsal cochlear nucleus very plastic. It has a high plasticity, and that's probably part of the problem actually. Next, the signal will be passed on to the midbrain, to the inferior colliculus, next up to the thalamus, and then we reach the primary auditory cortex, where we have a lot of processing of sound. And then, of course, connection with other parts of the cortex, and we are actually understanding what we're hearing. Here it's important to notice that there's actually more output from the primary auditory cortex than there is input. So this area is very important for the downstream feedback to the lower auditory system and regulating how we perceive sound. And now on the right side of the figure, you notice the hippocampus and the amygdala. This is also heavily linked into tinnitus. Hippocampus obviously with memories, and amygdala with anger and frustration, perhaps, like this limbic part of tinnitus where we feel frustrated and we're worried about it and we remember how bad it was another time and so on. And unfortunately, higher drive in this system has shown to actually worsen tinnitus. You have more neuroendocrine stress levels and so on. So it's a complex system, but still I have chosen my two focus areas, and that's the cochlear nucleus, and now recently I'm focusing almost only on the primary auditory cortex, because I think that's the places where we have a chance to actually figure out something. So first, looking at the dorsal cochlear nucleus, this is also from that same review, it is known that a lot of different changes are happening after noise exposure. For example, loud noise will actually make the synaptic terminals swell a little bit, and then they have a hard time connecting back to their postsynaptic densities. So we actually have fewer auditory nerve fibers that are functional after noise trauma. And this leads to intrinsic cellular changes and synaptic changes. So we might have an alteration in voltage-gated potassium channels or hyperpolarization activated channels dealing with excitability of neurons. There has been shown changes in NMDR receptors, so our glutamatergic receptors that are also heavily linked to synaptic plasticity. And then it's been shown that there's an increased glutamatergic transmission in this area, this first area of the brain, and a decrease in inhibitory transmission. So we have both GABA and glycine active in the brainstem. And together, this has been shown to lead to changes in spike time dependent plasticity. It can lead to increased synchrony. So we have a stronger output of a certain signal, or increased spontaneous firing rate has been measured. And all of these ones are part of creating tinnitus. Obviously, these changes can be further relayed to different areas of the ascending auditory system, but this is really where they've shown that the first changes take place. So then my lab's research questions, since we kind of know that the dorsal cochlear nucleus is the origin of tinnitus, we're just wondering, are the mechanisms creating tinnitus and the same as the one that gives us the perception of tinnitus? That has not been very well studied, if they're exactly the same type of neural activity that's part of creating it and continuing the perception of it. And then the second question that we want to address is also, obviously, how can we remove or alleviate tinnitus sensation? That's clearly the goal of everyone studying tinnitus, I think. But we're trying to see how we, in our approach, looking at neural activity and especially looking at this on a subtype level of cells is where we think we can contribute. So yeah, neuronal activity and what cells are driving these different phenomena of phantom sounds. So starting with the first question, are the mechanisms creating tinnitus and perception of tinnitus the same? So first we want to know then, how can we control neuronal activity to study this? So this was recently, we pre-printed this in bioarchives actually, because we were in a hurry, so Tawan Malfatti could apply for a postdoc. So this is his work from the beginning of his PhD, and we're now putting this out as using cortical neuron markers to target cells in the dorsal cochlear nucleus. So what Tawan did here was that he evaluated what kind of genetic promoters we could use in the DCN. And one classic one for the cortex to target excitatory neurons is the calcium calmodulin kinase 2 alpha promoter. This one is also used for targeting neurons in the hippocampus, for example. We have previously done several studies using another marker that's inhibitory, which is the nicotinic acetylcholine receptor, also 2-alpha actually, subunit. And he, in this first paper, he has compared them and seen if he can control and drive different populations of neurons in the dorsal cochlear nucleus using these tools. So he has injected virus with the CAM kinase promoter to drive expression of channel rhodopsin or arcurodopsin. So these are synthetic proteins used in optogenetics. Channel rhodopsin can excite neurons, and arcurodopsin can inhibit neurons. And he also used transgenic mice, Cranate2 Cree mice, and injected Cree-dependent virus to express channel rhodopsin in these animals. Here today, I will only talk about the CAM kinase data, just out of time constraints. So what he did, first of all, just to see if the promoter is expressed in cells we're expecting, we wanted to just excite them. So that's when we used the channel rhodopsin protein. So here you can see a picture actually in A of a mouse in a stereotaxic frame, and you can see the probe in the head in green. This is a single channel 16-channel electrode for extracellular recordings. And in white, you can kind of see an angular entrance of the optic fiber. So we stimulate with blue light into the DCN. And you can see some expression here since the channel rhodopsin is also co-expressing enhanced yellow fluorescent protein. And we did not see this like we expected to see in some kind of focused excitatory cell area. We saw it a little bit more widely spread. And in red, you can see the probe, the electrode that was dipped in color. But we saw nice membrane expression of channel rhodopsin. So we were thinking this is probably going to work. So what kind of activity can we evoke? So here's an example. When you see things in pink reddish, this is actually stimulation by sound. So we just look at units responding to sound. These are peristimulus time histograms, and you can see that after sound clicks, we can see a little bump in activity. And in the middle example, you can see a unit that did not respond anything extra to the sound clicks, but to blue light, it actually increased its firing. So we could see optogenetic driving of this particular unit. And in D, we have a case of a unit that responds a bit delayed to sound, but also very similarly delayed to light activation. So first we were thinking, okay, good, we can drive some cells with that. But then when we started to look at population data here in E in the bottom, you can see that baseline activity compared to sound activity in S, we saw an increased activity, which is normal. Comparing baseline activity to light, we saw that light also could increase spiking, but we had much fewer units responding to light. But then when we look at baseline activity compared to sound plus light, we actually saw a little bit less of activity. and comparing sound to sound plus light also showed less activity. So when we have sound and light at the same time, this excessive excitation actually contributes to lateral inhibition. So this system becomes extremely sensitive to sound when we're stimulating with channel rhodopsin. And this was not what we expected. So this makes experiments that you have to be very careful, you know, when you're using light in the auditory system to stimulate. So we were not completely happy, but we were happy that we could drive certain units. Our real goal was actually to decrease activity, since I already showed you that there's an increased firing going on in the DCN in tinnitus. So we wanted to see if we can decrease activity, so using the arteriorodopsin, which is a green light activated outward proton pump. Again, here we managed to pick out some really nice units that are very typical for the sound circuit. The circuit has onset to sound, so you see this very nice firing straight after a sound pulse. And with 2 examples of that, and then in example C, you can see a pauser cell, so it actually makes a pause in firing in response to sound. These are, again, kind of typical responses from auditory units here. But what happened when we applied green light in these experiments? So we are now expecting to inhibit these units to have no firing. Instead, we saw delayed firing. In all these 3 cases, we could see a very typical delay. So it seems like we're dealing with a polysynaptic circuit with, I don't know, inhibition, disinhibition, whatever going on. So again, not exactly what we were expecting, this delay. But this was interesting because in the bottom, you can see on figure F that we were looking at responding neurons, units that responds to sound, and they were slow firing, but we had a lot of non-responding units that we managed to pick up. So we wanted to see, are the non-responding units also somehow affected by this circuit? And then we saw that using the CAM kinase 2 promoter, here we could actually affect these non-responding units that either had a baseline firing kind of high or no baseline firing almost. When we applied green light to inhibit CAM kinase 2 positive neurons, we could actually turn a cell firing a lot to inhibit it, or we can have a cell firing little to turn on. So again, disinhibitory circuits. So we are clearly able to affect a lot of activity in the DCN using the CAM kinase promoter. So we were kind of like, yeah, maybe we should just consider that we have a tool that affects more neurons than we thought. It's not specific for glutamatergic cells, but we can most likely play with the excitation inhibition ratio here in some way, because we can inhibit a lot of spontaneous activity, for example. But then this only becomes interesting if we're looking if we can actually affect tinnitus perception, right? So that will be the next step. And here we just decided to, let's just switch tools first, because yes, optogenetics is nice. You can correlate with time to your responses, but it's not very practical in vivo, you know, with behaving animals. It's okay if you have them anesthetized and so on. But for behaving animals, we want to have a better tool. So then we switch to chemogenetics, obviously. And here we are using instead viral vectors to inject genes of membrane proteins that are G protein coupled. And there's 2 main types, the DQ and the DI membrane proteins. They are I think someone is speaking in the background. So these are muscarinic receptors that have been mutated, and instead of binding acetylcholine, they now bind a synthetic drug called CNO, which stands for clozapine N-oxide. And it's actually been shown recently in the literature that it's actually the clozapine that gives the effect to these receptors, but in very, very low dose, they're very sensitive to it. But here we decided to use the HM4DI protein, since it leads to hyperpolarization, because we want to lower the activity in the dorsal cochlear nucleus. So here Tawan went on to do a second manuscript. This is still unpublished. We're finalizing analysis to submit. But he did a very nice experimental timeline here. It's very tedious, and he has had mice that go through all these different methodological steps to get the data. And he managed to have, I think, a total of 18 mice survive all different stages here and produce very robust data. So first, obviously, we're looking at in vivo behaving mice because we want to study tinnitus perception. So for that, we need a tinnitus test. So here to the left in the bottom, you can see a schematic of the tinnitus test that was developed in 2006. It's called gap detection. So the idea of this test is that you have a background sound illustrated by this gray bar, and then you have a loud pulse. So it might be sound pulse. So it might be this pa. So when you have a pa, this mouse will startle. This is the acoustic startle reflex that rodents have. This is a kind of loud sound pulse. Then as a warning cue for them to not startle, we have actually a silence. So you have this, and then you have a very short, complete silence, and then you have this loud pulse. Then this works as a warning cue so the animal startles less. And then the rationale is, if you have tinnitus of a particular frequency that matches your background noise level, then tinnitus will sort of fill the gap. So they are basically not picking up this silence as well anymore, so they would startle more. And this has been shown by a lot of researchers that it's working. Not all animals can detect the gap from the start. That's why we need to do a pre-screening to exclude them. And here we excluded, I think, 4 out of 34 animals that we had in the beginning. So when they have been screened, they can detect the gap. These mice have been injected with adeno-associated viral vectors for the CAM kinase HMD4I receptor expression. So this is the inhibitory chemogenetic tool. Tawan waited about 30 days for having enough expression in the DCN, and then he did a hearing test of these mice, and then they were anesthetized, and he did auditory brainstem responses day 35 to see that they have normal hearing, because that's obviously also important. And then a few days later, he exposed his mice to noise, loud noise for 1 hour, 90 decibels, and then left the animals for 2 hours in silence. This has been shown by others that this helps to induce noise-induced tinnitus. Then he repeats the hearing test because we want to make sure we're not causing hearing loss, because we believe that there are different mechanisms behind hearing loss and noise-induced tinnitus. And we could see that there was no hearing loss, and then lastly, performing the tinnitus test again. So here you can just see an example of the, so there's a vibration sensor under this tube. So when the mouse startles, you get a current, and then you can plot this and see that there's what you measure is actually the suppression of the startle. So in yellow or orange in the bottom, you can see that animals after noise exposure were not as good at suppressing the startle. So all the orange bars are a little bit lower than the bluish bars, but it doesn't seem like any particular frequency is the most affected. And this has also been shown before in literature, that tinnitus is something individual, like different animals get it at different frequency intervals. But what we do then is we actually have to pick the most affected frequency. And when we plot the most affected frequency band, so this is the background frequency that we're trying to match the tinnitus, then we can see a significant worsening of the ability to suppress the startle, and this indicates tinnitus. So then we had all these animals that presented tinnitus, and then we wanted to see if acutely decreasing the activity of this population of neurons in the DCN could improve tinnitus. So in control, we have saline injections, then the animals on this consecutive day have CNO injections. We had two groups of animals, the ones that had the chemogenetic receptors and the ones that just had a control expression of enhanced yellow fluorescent protein. And then comparing the suppression index before, in blue, to after the noise trauma. You see that they're very poor at suppressing the startle. But then after giving CNO, these animals improves actually their ability to suppress the startle, indicating that they can better pick out the silence in noise and actually have less tinnitus. So we saw that for the animals expressing HM4DI, but not for the control animals. So we were kind of excited and saw that, wow, decreasing the activity acutely can improve tinnitus. But we wanted to make sure that we actually had a real effect. So we also did unit recordings in these mice just to show that indeed CNO had an effect. And I'm not going to go so deep into that, but we compared firing rate, tuning broadness, and best frequency. And we saw that for these animals expressing the chemogenetic receptors, there was a decrease in firing rate, a little decrease in broadness of response, and a little increase in the best frequency they respond to, and none of that was seen in the control animals. So we were like, yeah, we're really seeing an effect here in behavior based on firing activity in these neurons. But then we wanted to know, can we then just block tinnitus induction if we apply CNO during the noise exposure? So we tried that. We did exactly the same that I already described to you, but with the difference that we applied CNO during noise exposure. And cutting it short, there was no difference here. We saw the same kind of results. The animals still presented tinnitus-like behavior. There was no hearing loss. We just created again some kind of noise-induced tinnitus. But then what was interesting was that when we tried to rescue this 5 days later, when we wanted to see if we could also improve, have them notice less tinnitus, that didn't work in these animals. Somehow, like the CNO had already blocked the activity of cells that we would later be able to decrease the activity of. That's how we kind of interpret it, because we couldn't see this rescue effect anymore, indicating that there are different populations involved in causing tinnitus and maintaining tinnitus. So if you just look at the conclusion here of this study, is that we acutely inhibiting only works for this mouse that weren't already, these cells weren't already inhibited during a noise trauma. And the only difference that pops out immediately is the firing rate of the units that we are affecting. So if you look in D, you can see in one case we have a lot of units firing between 50 and 100 hertz, while in the second case we only have units with an average firing of 20 hertz. So it's like we have some kind of high firing rate population that is not present in this second scenario. And this is now what Taiwan is analyzing in detail, correlating these units, firing rate, broadness, and best frequency, and trying to figure out if there's a particular type of unit that is not, that is the one that can help reduce tinnitus perception. So I'm going to have to leave you with a little bit of cliffhanger for that particular talk, but we are very excited about this work. So then for the second part, which is shorter than my first part of the talk, how can we remove or alleviate tinnitus sensation for real, I was going to say. Well, going back to this transcranial magnetic stimulation, the one that showed possible efficacy in humans. now it would make sense, right, to try to stimulate the dorsal cochlear nucleus. But unfortunately for humans, that's an unpractical region to stimulate, because maybe we can cause vertigo, or we can mess with other areas in the brainstem. It's a very, very small structure, and we have our breathing centers in the brainstem. I mean, who knows, you know? It's not like the first area we're thinking of manipulating. So instead, most of these studies where we try to decrease neuronal activity is aiming for the cortex. So another problem for studying this is also the size of these coils. When we have these coils, the amount of loops and the size of them will help to create a certain strength of a magnetic field. The same apparatus doesn't really work in rats and mouse. It becomes ginormous compared to the human skull. So it's much harder to focus the stimulation. So this is a bit tricky. But basically, simplifying it a lot, how transcranial magnetic stimulation works is that we pulsing current to these coils, and then each current will actually generate a magnetic field. Then through electromagnetic induction, which is not my strength, this turns into an electrical field in the neuronal tissue, perpendicular to where these coils were. And as you know, if we create an electric field, we have like volts per meter, which is usually how they measure it. Then we're able to affect voltage-gated ion channels. Obviously, we can open them and we can create current flow and so on. And then we're dealing with possible excitation, who knows, maybe also inhibition of certain neurons that are receptive to this stimulation. This is not very well known exactly how what effects transcranial magnetic stimulation have on the neurons. But what we do know is that the magnetic field, or the electrical field, I should say, decrease with distance from the cortex. So you have the strongest effect on the very, very surface of the brain. And that's interesting because there's a lot of specific things going on the cortex surface. Layer 1, I should say. So at Instituto do Cerebro, we have this policy of everything is doable and we can make everything ourselves. So of course, we were like, we want to try this and let's build our own TMS coil. So George Nascimento, a collaborator who's fantastic, he started to making coils and measuring the magnetic field we could generate, and then he made a more specific one to be used on mice, smaller with an angle, so we can actually focus the field better, like trying to hit the smaller area. And we've been testing the strength of this, and we have very few pilot experiments actually done in mice, which you can see in this picture. But we were very excited to see that just using this prototype, and we did stimulation for about 20 minutes, but it wasn't very high frequency because we still have to figure out how to deal with heating, excess heating. So we're stimulating a little, and we're planning to apply some insulators in between here, but to take part of the extra, wow, that I don't know how that happened. Can you see some red drawing on my screen? Oh, if not, I don't know, it just appeared. But what we could see is...
you can see the drawing.
Yeah, where did it come from?
This must be the kids, no?
I'm sitting here alone in the office.
The kids in the pandemic. Really
yeah.
nice. It doesn't bother.
Yeah. No, look, I didn't touch it. Someone is... Here's my hands, and it's increasing. That's funny. So
there's instruction that you should do a Control Z or a click ask. I don't know. The in the chat people are saying, I don't
know. Yeah. Okay. It's gone. Close. it's amazing how there's always some kind of new drama every time you do a presentation. That was very new to me. Let's see again. Sharing. And so we're back, I think.
Yeah.
All fine.
Mm-hmm.
Yes.
We're back.
Ghost in the computer. So we were very excited to see that we actually had some CFOS activity after using our home-built TMS for mice. So you can see some grain cells in layer 2, I presume, of the cortex, and we have a lot of students helping out with this, like one student helping to put the mouse under anesthesia, and then George came and with his huge batteries to be able to generate these current pulses. And then we had another student doing the perfusion, another one cutting it, and someone else imaging it. It was like this. We were all very excited. But yeah, we have some proof that this might work. But now the question is, what cells are we actually wanting to manipulate? So now, super quickly, just run you through 3 images from some work by Ingrid Nogueira, a very talented master student who's now starting her PhD with me. So she started this thing of seeing if are all the pyramidal cells of layer 5 affected the same way by noise exposure. So this was a kind of a simplified experiment, not at all as the previous one. She did noise exposure and then 7 days later sacrificed the mice and cut coronal sections to use whole cell patch clamp and record electrical properties of these neurons. So here we aimed for layer 5 because this is the output layer of the cortex. And then she divided them into type A or type B cells based on electrophysiological signatures. So you can see in this example that there's an after undershoot and an after overshoot when you're doing train of action potentials or a hyperpolarization. This is not seen in type B cells, so this is like ion channel dependent features, and they are correlated. So the type A signature is correlated with corticofugal cells that goes out down from the cortex, and type B cells are more telencephalic connections, probably going to the contralateral primary auditory cortex, for example. And then we took help from our statistician to do some principal component analysis of, I think it was 16 electrophysiological features, and divided them in type A and type B types of cells, and we could see that they kind of clustered differently, the variability of them. So light blue is control A cells and dark blue is control B cells. And then when we compared type A and type B cells from noise exposed animals, we saw that they kind of spread out, their variability spread out, but like in different directions. And when we added a third principal component analysis just to illustrate this variability, we twisted it here in a 3D angle, so you can see that there is kind of a good separation. But what's interesting is that noise exposed cells appear to be more different, like there seem to be more variability, they have different features. It was hard to find any particular electrophysiological property that was distinctly different except for firing frequency. The other ones had kind of trends of being different, but nothing was significantly different. And this is data from I think a total of 108 cells. But here we found that if you make these cells to fire in response to just a current injection, and then looking at the initial firing frequency, the first 2 action potentials, and what we call steady state frequency, the last 3, we can see that there's a difference between type A and type B cells, and also for noise exposed and controls. So what pops out here is that for initial frequency, if you look at the light blue line, this is control A's, they decrease their initial firing frequency after noise exposure. This was not seen for type B cells. But for type B cells, we instead noticed that steady state frequency, now comparing dark blue with dark red, increased a lot in noise exposed animals. And here again, the type A cells decrease firing. So this was the same was seen depending on different current injections. You can see in the bar graphs in the bottom, but basically type A cells decrease their firing frequency and type B cells increase. So there's some kind of opposite direction here going on. This was interesting. We tried to see if this was part of like the gain, if each cell where you inject more current will fire faster. That's standard. But we would just wanted to see if some of them increase this a lot or not. But the only difference we saw when comparing the gain, which is basically the slope here of frequency and the current injection, was that after noise, the steady state gain becomes very different between the type A and type B cells. So they're just responding, one slower, one faster. But then we wanted to see, does this hold up in vivo? So this was actually in collaboration with Jessica Vinny and Hichardsson Leão. So they are experts in miniscopes. So Jessica is a super talented PhD student of Hichardsson, and she is really good at implanting these lenses, and in some cases also a little prism. So you implant that into the brain and kind of create this tiny little window where you can image cells that express genetically encoded calcium dye, for example, GCAMPs. And then the miniscope, which is a miniaturized microscope, super lightweight, you actually connect onto the head when you're doing some behavioral analysis, for example. And then you take this off when the mouse is just going back to its home cage and so on. But what we saw in this preparation, this is just an example, was that indeed we can image distinct neurons in layer 5, and then we can submit this animal to this noise exposure, and then 7 days later put back the miniscope and image the same cells again. So you can actually find them based on morphological landmarks and using statistical programs to prove that this cell is indeed the same cell that we imaged 1 week before. And here's just an example. Control cell number 1 firing kind of regularly in response to tones or sound clicks, actually. And 1 week after this noise exposure, the same as in Ingrid's experiments, we can see that that cell fires much more. And in 2, you see a cell that fires not so, that fires kind of normally will fire less, the red trace in the bottom, after this noise trauma. So it's just 2 simple examples showing that this is possibly also happening in vivo. So we have some kind of dual ways of regulating neuronal activity after noise trauma. So just to sum up here, this was or is kind of in contrast to what has been published before. This leads to a bit of like, how are we going to understand this now? Because this was published in 2018. They use a similar noise exposure as us, just a little bit louder. And they also focus on layer 5 pyramidal cells. They focus on corticofugal projecting neurons, which is what we call type A cells. And they saw here using head-fixed mice and 2-photon microscopy. And while they were also doing measurements of the auditory nerve, they could see that after their noise exposure on day 1, they will have less auditory nerve input, which is classical seen. But then for the type A cell, they saw that the responses of calcium transients of the neurons from layer 5 of the cortex going to the inferior colliculus, they were increased. So there was some kind of increased feedback to this area after noise exposure. And this also persisted for 7 days. So here we're like, but we see our type A cells to fire less. And they see that they fire kind of generate a bigger response. So what's going on here? So just to finalize this now, what is important to understand here also when we want to affect cortical neurons is that subtypes of pyramidal neurons are controlled sometimes by different interneurons. So this was actually shown by Marcus Hilscher, my first PhD student a few years back, who focused on Martinotti cells. This is this interneuron of layer 5 with this beautiful axon going all the way to layer 1, where it heavily innervates the distal dendrites of layer 5 pyramidal cells. And what he found was that it specifically aimed for type A cells and kind of bypassed type B cells. And this is unexpected, but there's some kind of specificity to these large type A pyramidal cells. And then when he stimulated Martnotti cells, like a whole group of them with optogenetics in some kind of beta frequency, he saw that if he patched 2 pyramidal cells that were spiking out of phase to each other, when he applied light to stimulate the Martnotti cells, they became synchronous. So even then they were firing sparsely, they would fire more synchronous due to the Martnotti cell activity. And then he showed in this paper further the mechanisms behind this. So that's interesting. So then Ingrid has now started to look at the Martnotti cells in these noise-exposed animals, and she sees just in some preliminary data that indeed it appears like the Martnotti cell is more active after noise exposure. They need less current to fire first action potential, and they have an action potential threshold closer to resting membrane potential. So in that sense, we're now wondering, because we made this little model, which you can see a very simplified drawing to the right, where we have the type B cells increasing firing going probably contralaterally, which could be more contralateral excitation, probably keeping the tinnitus going maybe. And the type A cells that we thought would have less feedback because they had decreased firing frequency. But now adding this idea of Martnotti cells, maybe even though they fire less, they might be more synchronous. So they might drive a stronger feedback output. So this is something we have to study further. So conclusions of study 2 then was that noise exposure increases type B pyramidal neuron firing and lowers type A. As I said, it might increase intrahemispheric firing, but then we don't know if it decreases or what does it do with the feedback. You know, do we have overactive Martnotti cells here? So obviously, a new hypothesis would be to look if noise exposure can synchronize type A pyramidal cells and give a more powerful output. And here we should obviously use miniscopes to see if you can see more cells blinking at the same time, for example. And the take-home message for this talk would be that noise-induced tinnitus without hearing loss can be generated in mice and tested with behavioral startle tests. This is nothing new, but we are very excited that it works in our lab. And that there appears to be different mechanisms for generating and maintaining tinnitus sensation. So how then can we remove this sensation? How can we add some kind of inhibitory feedback? This is the trick, since the feedback is excitatory. Can we maybe create long-term depression somehow? Like, these are ideas that pop out. And then we've shown that the auditory cortex is affected by noise-induced tinnitus non-uniformly, and that might have something to do with Martnotti cells. So next, we want to know then, can TMS alter Martnotti cell activity, for example? Because as I said, the magnetic field or the electrical field will be strongest in the very, very surface. And what do we have in the surface? We have the axon terminals of the Martnotti cells. So maybe we can modify their responses. And in the long run, we are hoping to restore normal homeostatic plasticity, obviously. So I just want to thank all the students and staff of Instituto Cerbo. It's a fantastic place to work, and we are so lucky that we have our brand new building that we're all starting to move into now. So please come and visit us at UFRN in the future. And I'm very happy to have sponsoring from the American Tinnitus Association, Hippocampus, of course. And then I would like to thank my lab, the Hearing and Neural Activity Lab, and the neurodynamics units that we collaborate closely with. And a special thanks to George with the hats you can see, who built the TMS for us. And thank you to you all for listening. There.
Wow. Amazing work. Thank you. Yeah. Really amazing. We've got a lot going on.
Yeah.
It seems incredibly complicated, but
yeah.
fantastic. Since it is very hard to actually think on to think of a tool that can regulate specific cell populations, and at the same time with a broad kind of stimulus, it's really, I think that your approach of looking at the populations that are involved and how they are how they are working, right, is incredible. But you think that that it is actually possible, or how could you modulate this? And actually, Claudia Vargas is asking, which a specialist in TMS, if you expect tinnitus and startle reflex response might share neuronal circuits in the brain.
Yeah, this is a great question. It's actually a colleague of mine here in Sweden. That's why I'm here in Sweden, to collaborate with him. He has shown that it's not exactly the same mechanisms involved in PPI and this gap detection startle. So that I can answer. So normal pre-pulse inhibition is a slightly different mechanism than this gap-induced pre-pulse inhibition. So this gap is measured by neurons in the auditory cortex quite specifically. So that's going on. And then the question is whether tinnitus will affect, what was the specific question?
Excuse me? Claudia's question, you mean?
Yes, please.
Maybe she can appear. Claudia, can you show up here so that Kia can see you?
Because if the question was...
There she is. I don't see
you
her.
for a very nice talk. My question is if you expect that tinnitus and startle reflex response to share similar brain circuits.
Right.
How to disentangle these two circuits if they are different? So.
Yeah, no, so now I remembered why I was starting to say this. Obviously, if the auditory cortex is part of the startle response, then it might share it, but it's like a different pathway going from different nuclei that deals with actual startle reflex. That's more complicated than just the auditory part. So maybe the end point of it in the auditory cortex, but then the others, I don't know. But since people are using this as a test for tinnitus, it would be terrible if it's actually the reflex that's worse and not the detection of it, for example. So I think with the amount of literature, I don't think it's exactly the same circuits, no. I think one can be used to test the other.
Okay, thank you.
So I was wondering, you said also that tinnitus leads to depression and anxiety and depression. And my question is whether it could be the opposite, actually, if depression and anxiety could lead to tinnitus, or if there's some evidence in that regard.
Yeah, I think that can for sure contribute to the severity of tinnitus, but it's been shown that there is higher neuroendocrine like stress hormones, higher levels in people with tinnitus. But then if you have them from the beginning, if you're more vulnerable to tinnitus, there's a whole field of these kind of studies. I think there's presumably yes, but it might not be that super common. I don't think you get tinnitus from pure stress like that. You can get tinnitus from biting your jaw or like tensing your shoulders, but that's a different kind of tinnitus. So yes, you can get it from being extremely stressed, you know. Even I get that when I'm very stressed at work. Sometimes I get this like beep and I'm like, oh my God, please no. But I think those are a little bit different mechanisms. But for sure, how bothersome tinnitus is exaggerated by the stress. That's shown.
Okay. I don't see questions here.
Yeah, maybe I should.
Maybe I can ask
sharing so we can see the chat.
Right. I don't know if we're back in YouTube, but I was going back to my initial question, which is this stimulation, this global stimulation of the TMS. So how can you actually, how would be, I understand that maybe it's a synchronization. How would you expect this to work to actually get what you need and not disturb anything else? And is it lucky or what's that? I mean,
no, we know that one of the problems with the human studies is that they all use different frequencies and different polarities and different angles, so it's a kind of a mess. So what we are proposing in this grant application we wrote was to go through and test how different frequencies will affect different, for example, interneuron population. Also, if we can have low intensity, are we only capturing the very layer 1, for example? If we have higher intensity, how deep can we go? So this is exploratory studies that are needed to be done, and I think they would be very interesting to other areas of the cortex as well, for sure.
Yes.
And we are hoping that we can, in the long run, use miniscopes at the same time as TMS, because we don't have any electrical interference, because the miniscope is purely light that we're detecting. So we have all these ideas to test. But first, my idea was, since we have this previous study of the Martinotti cells, and we know that they are very sensitive to the stimulation frequency, I was hoping that we could maybe pulse in a certain frequency to activate them more than other cells, for example. But now, when the research is kind of showing that maybe the trouble is to have excess of Martinotti cell activity, then I'm like, oh, then I have to, how can I revert it then, you know? So looking more at how to induce long-term depression, how we can kind of down-regulate synaptic release, for example. So it's still early days, but I still wanted to share it with you because I'm so excited about it.
Yes, fantastic.
Yeah.
There's some more questions here. Brenda is asking, do you want to show up, Brenda, to ask your question? Can TMS lead to other brain effects, adverse effects as epilepsy?
Right. So, again, I'm not an expert on this. I've only read papers, but what's usually done for TMS is that you use a motor threshold to have an effect in a patient. So you stimulate in the motor area, and then you measure muscle output or a muscle EMG, you know, electromyogram, for example. And then people usually set 110% of the threshold. That means, in my mind, that you are stimulating all the way down to layer 5, the large pyramidal cells that will excite your muscles. So then we're dealing with kind of a high intensity.
Right.
I don't want to go all the way down there, you know. I want to have a lower intensity to just, in the beginning, affect the surface, like layer 1. There is 4 types of interneurons in layer 1, and I would like to study them one by one, actually. So
I see.
Epilepsy, yeah, if you push it too far in a vulnerable area, maybe. I'm not expert enough to say that, but I think a lot of studies always try, like, if we do it more, if we turn on the light more, we will have more optogenetic output. People just want to put more drugs and higher and higher. I think we should do the opposite. I think we should scale it back to where do we actually have a real response that's specific, you know, so lower.
Okay. Claudia wants to comment on that before I make Newton's question.
Yes, maybe I can give a little help. In humans, single pulse TMS in the primary motor cortex in general do not induce epileptic seizure unless if the person has some some signs of epilepsy. So this is why you have to be very careful in choosing the population you are going to test. See, on the contrary, when you use this paired pulse or multiple TMS pulses, these protocols where you apply several pulses through interval of time, this could be inducing epilepsy on even on people that didn't that had an epileptic behavior unnoticed until that point. Let's put it on this way.
Yeah.
Thank you, Claudia. Newton has a question. Newton, do you want to show up?
Oh, I might. Thanks. Let me see if I can get the camera. Hi, thank you very much for the talk. It was really beautiful work. You mentioned in one of your slides, there was something about tinnitus and auditory hallucinations. So it made me wonder if the gap detection is also affected in psychotic diseases, as is PPI, and if there is any connection between tinnitus and schizophrenia, for instance.
Yeah, that's something I would actually like to study a little bit more. I mean, PPI is classically used to detect psychotics, animal models of psychosis, of course. The gap detection is a little bit more tricky, and since it specifically involves the auditory cortex, I don't know if it's a very good model. I just heard a talk by David Nutt from the UK that studies psychedelics, for example, and he says that in LSD, for example, you don't have an auditory hallucination. That's kind of gone. So that made me think, like, how good is these kind of tests? I know a lot of people are trying to test this to see if it would be some kind of biomarker for psychosis, because they're trying to find it, but I don't really know this. I have another PhD student, and she's looking at sensory filtering, and she has also this very interesting data, but for another talk. And we're just looking at paired clicks. This has also been done for psychosis and for schizophrenia. So they have less sensory filtering, meaning that like double clicks will have equal neurological like response. Usually in healthy subjects, you have like a big response and then a lower response, because this is a repetition, it's not important. But for schizophrenia, they've shown that this is, the clicks generates equal amount of response. So here in her project, we are actually testing, first of all, animals that are having noise-induced tinnitus and not, and trying to compare this. But then we are also provoking the system with, we have tinnitus extracts. And in her case, we use this very high concentration of TMC, no, what is it called? THC, sorry, THC, that could evoke maybe some kind of psychotic stuff. So this is early analysis still. It's very hard to analyze this data, but yeah, we don't know, but I'm very interested in it. So if you have any comments or help for me, I would gladly receive it.
Well, no, I was just curious because, you know, it's sensory gating, and you know, the circuits are connected. Maybe there could be something like an inverse correlation between tinnitus and schizophrenia or something like that.
Yeah.
I just wonder if there is any epidemiological evidence. Maybe it's worth looking for in circuit basis.
Yeah.
Yeah, I know nothing about it. I'm just curious.
Yeah. The problem with the literature is that there's a lot of little studies here and there, but some are showing something, some are not showing it. So it's always a big contrast, and it's usually because in patient, the type of tinnitus that people have is different. So we're never looking at exact same models, but that's what we're hoping to be able to do with very robust mouse models.
Okay. Well, thank you very much. Congratulations again.
Thank you, Newton. Also, Rafael Linden wants to make a comment. Rafael, are you there? There he is.
Yes, it's not a comment, a question. I thank you for the presentation, Kia. What I would like to know is whether there are any genome-wide association or other genetic studies of risk factors for tinnitus.
So this is very much why I came to Sweden. So I'm here for as a visiting professor, and my colleague here, he's doing this GWAS, or genome-wide association studies. He has tried this. They published some work on tinnitus in twin studies and saw that there is a genetic component. But when they did this huge study of tinnitus patients not genetically related, nothing really came out. It's very hard. They didn't see anything. And colleagues at the Karolinska Institute, they have shown GWAS data for schizophrenia, and they're finding like markers. So he was very excited to see if we could find that for tinnitus, but so far it's a tough one. It's not, I mean, from the twin studies, we know there is a genetic component, but genes that increase risk factor, not really known yet. But I think there's a lot going on here, refining these studies. It's gonna come, but this is for these huge and rich labs of the Karolinska Institute to invest money in, because it's a very expensive technique so far. But yeah.
Thank you for the talk. It was really great.
Thank you.
Kia, I think we're done with the questions. And since our YouTube did not work in parallel today, so we're gonna put this talk later on there, if that's okay with you.
Yeah, that's
okay. Fine. Okay, thanks so much.
Thank you for the
invitation.
invitation.
I was really impressed. Very, very, very nice work. Thank you very much, Kia.
Thank you. Great.
Thanks a lot.
Great.
People got all the information about you in the email, so whoever wants to ask further questions, they can go for her email.
Yes,
Thank
and
you
then
very much.
Maybe next year I can come visit you guys instead.
Oh, great.
I would love to.
Yeah, that would be wonderful. We have to organize that.
Yes, great.
Lou wants to say something about the next talk, or next Monday is actually a holiday, right?
Yeah. Thank you, Kia. Então, eu queria agradecer a presença de todos, né, que participaram até agora com a gente. Semana que vem a gente não vai ter, porque a gente tem feriado, né, no dia 12, mas no dia 19 a gente tem a Carmen, da Universidade Federal do Rio Grande do Sul, que vai falar para a gente sobre o transtorno do aspecto autista, do fator de risco à neuroproteção, um caminho. Então, a gente se encontra agora no dia 19. Thank you, Kia.
Thank God, Frid.
Okay, I'm turning off here. Thanks a lot. Bye.
Bye.